I'm Marco Lobba. I'm the CEO and co-founder at Catena Bio, and I'm excited to share with you some of the latest updates from what we've been working on. Overall, I'll be walking through ADC production, particularly for the dual payload, how we build and consider safety, efficacy, how to build truly next-generation ADCs. And then I'll dive into the sister conjugation approach that we've developed here at Catena Bio, what we've been doing with it, and some of the most recent exciting data. So overall, we've seen amazing progress with ADCs, right? This is clearly a modality that is revolutionizing care for a large number of patients. Nonetheless, we do still see some failures, and even in late stage. And we've seen a lot of failures, particularly in lung, and that'll be a note that I keep coming back to in this presentation here. And overall, what we need, and it's become very clear, and I think Daniel's talk from Sutro earlier really outlined very well, we see that resistance to payloads is emerging as one of the major limiting factors in overall ADC efficacy. So our challenge here is how do we overcome that? Not only do we see generally poor sequencing capabilities for the same type of payload, we also see lower durability than we might like with even some of the very, very high-performing monopayload ADCs. And of the ADCs that have failed in lung cancer, they all have failed versus docetaxel, which is systemic tubulin therapy, which is something we think is quite interesting. And so overall, we think that this monopayload approach is a major challenge leading to a high amount of resistance, and that is typically drawn for that payload as a dominant factor. I'm not discounting the downregulation of certain tumor antigens, but we think payloads are definitely a leading cause of resistance. And we've seen very clearly that sequencing of similar payloads time after time is giving us reduced benefit. The other challenge in ADCs is for most of the ADCs in use today, we see that dose-limiting toxicities are typically systemic, non-target, not even on target off tumor. We see general systemic tox from free payload. And some of that can certainly be attributed to the conjugation strategy, how the chemistry of how that is attached. And so these two challenges here are side-by-side areas that we'd like to address with a next-generation antibody-drug conjugate. And so I'd like to set up or put forward some criteria we think about when we think about what an ideal ADC might have. We'd like to have multiple payloads to avoid single payload resistance. We'd like to have more stable conjugation, or at least tunable stability in our conjugation that will allow us to reduce dose-limiting off-target toxicities. We very clearly need to be generating homogeneous products. We can't have heterogeneous products. I think the general field has moved far away from that very quickly. It's very important if you're combining multiple different payloads to have a tunable DAR. You need to be able to match your potencies. And you must have a clear pathway to large-scale production of these types of assets. And ideally, you'd be able to accommodate a variety of next-generation payloads. We just saw an amazing talk on what some of the future of payload development might look like. And so, as we're thinking about payload selection, we think of two kind of key questions. One, synergy, I think, is an interesting term. I don't like throwing it up. I think really what we think about is 1 + 1 greater than 2 is kind of what we look for. We want to see greater potency than the individual payloads. You want to see addressing of clinical deficiencies for the monopayload ADCs. But there's some caveats here. Not all combinations are necessarily medically necessary. Can you get by with a systemic administration of one of those mechanisms is a common question that we ask. And again, as the presentation earlier showed, not all models are equally sensitive to both payload classes. And so you need to make sure you're interrogating a variety of different models to make sure that you're actually getting an assessment for the contribution of each payload class. And so for us, that's how we think about optimal payload selection, while also being sure to screen for optimal DAR of the two payloads of interest. And then from the other side, there's orthogonality. There's this question of, are you targeting different pathways? Our approach to combination selection is we'd like to target as orthogonal of pathways as possible, making sure that if there is a compensatory resistance that occurs, you are maximally protected from any individual shifts in overall cell cycle, cell process. Again, this is not necessarily meant to be a industry-wide limit. This is just how we at Catena think about payloads when we're selecting them and for optimal combination. One thing that I think has been really interesting when we think about payload selection, there's a lot of work on the DDR space, and there's been some recent publications by Bardia's lab over at UCLA where they've been showing that they can actually combine PARP inhibitors with sacituzumab govitecan treatment, and they show that by just delaying administration of that PARP inhibitor, they are able to completely eliminate any of the combination DLTs by just waiting about 72 hours before administration of PARP inhibitor. And so this is the kind of stuff where, again, we like to ask the question, is there a medical need to have these on the same molecule, or can you get by with a co-dosing strategy? And I think this data is actually really exciting because it points to some potential futures when you think about combination therapies. What can you combine with some of these ADCs to improve activity, improve potency? A lot of the work that had previously gone into that was based off of irinotecan systemic chemotherapy, and that was very much intolerable. But now with ADCs, we're actually seeing that there is a pretty good tolerability for PARP inhibitor administration very shortly after ADC administration. So I think that's a really interesting data set that's been emerging just in the last year here. And so as we look at how we can build ADCs, there's a lot of approaches that have been explored. You have combined conjugation, which often suffers from some of the weaknesses of the historic legacy conjugation technologies out there. There are branch linkers that I think have some really interesting data, and we saw earlier today some fantastic data from the artificial amino acid approach out there. And then there's been a variety of approaches looking at different enzymatic conjugation strategies to attach multiple different payloads. And unfortunately, the typically high amount of enzyme that's required for these enzymatic conjugations has been a major barrier to large-scale production. And that's an area where we'd really like to advance and hopefully significantly reduce the amount of enzyme and the number of enzymes that have to be purified from your final product. So again, we want multiple payloads, stable conjugation, site-specific conjugation, tunable DARs, a manufacturable process, and ideally a broad flexibility on the types of payloads accessible. So here at Catena, we have developed a whole system that allows us to build what we think are some very exciting molecules quite rapidly. We have core capabilities around enzymatic conjugation, highly stable conjugation, proprietary chemistry. We have computationally guided design, and we've turned that into this ability to attach multiple payloads, regardless of size and complexity, to a single IgG, with very, very precise control of conjugation site, phenomenal stability, very, very mild reaction conditions. And all of this, because we link two native amino acids together, tyrosine and cysteine, we have an incredibly broad substrate and modality capabilities. So while we'll focus today on that top box, which is protein payload conjugates, but we also have significant data around protein-protein conjugates, protein viral capsid, protein oligo, and even protein cell conjugation as well. So the core of Catena is this reaction. What we do is we have this enzyme, we call it catenase. It oxidizes tyrosines with a very, very high level of selectivity. This enzyme has evolved a deep active site explicitly to avoid modifying surface tyrosines on proteins. It's cytosolically expressed. You can imagine that off-target oxidation might lead to some negative cellular repercussions. And so we've been able to take this enzyme and modify it and use that beautiful selectivity that nature started with and expand to some very selective modification of specific tyrosines in certain sequence contexts. What that lets us do is be very precise on our modification. We don't touch tyrosines in CDRs, where you see many, many tyrosines. We don't touch tyrosines on the surface of most proteins. We only modify tyrosines at our target sites. There's one natively on IgGs, and we can install many more in different loops where we'd like them to be. This process is incredibly efficient. We use less than 1% of enzyme relative to the mass of our antibodies, and in our optimized work, it's far less than 1%, so it makes cleanup much, much more simple than other enzymatic conjugations. And you'll notice from this chart, you see that that enzyme is intentionally drawn only involved in this activation step. Because our enzyme does not actually form that CY bond, we don't see any reversion of the product once it's formed. And so our yields are extraordinarily high because we don't have to worry about breakdown of that final material. And again, because it's two native amino acids, tyrosine and cysteine, we have an incredibly broad set of modalities that this technology can enable. Today is just going to focus on the ADC application. So the question that we often get is, how do we use this one enzymatic conjugation and the same chemistry to attach different payloads at different sites? Again, we're gifted a very high steric sensitivity of this enzyme. And so by modulating the reaction conditions, under our first reaction condition, we're able to prevent access to the sites near the hinge region by modulating the flexibility of the IgG. There's a lot of ways you can do this that have been published on. I won't be disclosing exactly what we use here at Catena today, but there's been lots of publication on how you might decrease the flexibility of an IgG, lots of formulation studies on this. We use very mild changes in reaction condition to block off the sites near the hinge region, and then under our second reaction, we're able to then attach that secondary payload. The end result of this is absolute control over the sites of modification, which lets us adjust our DAR while still delivering a homogeneous product using a single chemical conjugation approach. This for us is, I think, a really transformative capability and something that we've been very excited to see how beneficial this approach is in generating superior molecules. So overall, this technology, I think, addresses some of the two key design parameters that I mentioned earlier. We have the ability to attach these multiple payloads with precise DAR, and we've selected topo-1 and tubulin inhibitors to go after, again, that key challenge in the lung space. We've seen, again, lung, particularly for NSCLC, when topo-1 fails, it fails versus systemic tubulin inhibition, and so that's why we're very focused on this particular combination for the indications that we're interested in today and for the targets that we're interested in today. So I'll be talking about our TROP2 targeted lead asset primarily, but we have a number of other assets in development. Overall, we do see superior activity for that combination approach, which is really exciting. I'll dive deeper into that data shortly. We also do see superior stability of this conjugation approach, which we've seen translates to significant reduction in off-target toxicity in a variety of our early mouse and non-human primate studies, overall giving us what we think is a really exciting molecule. So again, this gives us dual payloads, superior stability with tunable DAR, very strong manufacturing, and some really exciting data on, efficacy and safety. So I'll start with a proof of concept molecule. This was actually a HER2 targeted asset. We wanted to build head-to-head versus Enhertu, which we think is one of the most well-performing ADCs in the market today. And in their original studies, the Daiichi team showed that in this JMT1 model, which is low responsive to topo-1 inhibition, they showed that 10 mg per kg dosing of Enhertu led to tumor stasis for out to day 25. And so we took that same model and we ran a similar comparison here, dosing at 4 mg per kilogram instead of 10. And what we see is that with this combination approach, 2 tubulin, 4 topo-1 inhibitors, we are able to fully eliminate in this JMT1 tumor model. Now, it should be noted that the JMT1 model is particularly sensitive to auristatin combinations. The speaker coming after me, I'm sure will be telling you more about that because Pfizer's done a lot and Seagen have done a lot of work looking at this. And so we wanted to investigate whether this continued to be true across a variety of cell models. And so I'll now dive into the data that we've generated with what has become our lead asset, CATB101, which is a TROP2 targeted version of this multi-payload ADC. And so here we're looking at the NAD7 gastric tumor model. And the reason we like this one is that when we look at the previously published data by other groups, LegoChem in particular has done a lot of work with this model, and they've showed that their auristatin-based ADCs require significant dosing to yield tumor regression. And so here we see this one as not exactly auristatin resistant, but much less sensitive to the auristatin component of these ADCs. And so here we went head-to-head versus Trodelvy, and we showed that Trodelvy does lead to eventual progression, although it is significantly reducing the overall tumor growth. In contrast, with 1 mg per kilogram of CAP-B101, we're able to generate sustained tumor elimination for the lifetime of these mice. So we ran this study out past day 80, out to day 80, and we did not see any significant tumor regrowth, showing that not only are we cutting down tumor growth, we are actually completely eliminating these tumors from these mice at extremely low doses. We then ran the study, the follow-on study, to examine what would happen if we redosed that Trodelvy-treated group. And so half the mice received a second injection of Trodelvy, which led to rapid tumor regrowth. And the other half of the mice in the Trodelvy-treated group received an injection of CAP-B101. And gratifyingly, we do see significant tumor reversion down to below starting tumor volumes. The study had a pre-specified endpoint of 80 days, so we didn't go beyond that point. But this overall was, for us, really exciting and really validating that this combination approach is generating a really, really effective molecule. The next study we did was back in that JMT1 model, and here we ran a head-to-head to the other topo-1-based ADCs on the market today, Trodelvy, Datroway, and HER2. And we see a very, very strong, again, improvement in overall efficacy when dosed at the same level of these leading ADCs. And when we reduce our dose to 1/4 of that concentration, to 1 mg per kilogram in this hard-to-treat tumor model, we see that we're able to meet or beat some of these approved ADCs for overall activity, showing that we have a sustained significant improvement in overall potency. So this was really exciting. And what's even more exciting is the recent data that we've generated in the PDX setting. So the challenge, as Daniel mentioned earlier, in measuring combination activity in a CDX model is you have a monoclonal group of cells in that mouse. It's very hard to see how that combination makes an impact in such a homogeneous setting. It's the heterogeneous setting that is a real-world setting where you actually are going to see combinations of efficacy from your different payloads. And so what we did here is we looked at a naive PDX model where we showed that with a single injection of Trodelvy, there is a very robust sensitivity to that topo-1 inhibitor. We're seeing quite a strong response here. I mean, this is a very low dose of Trodelvy, far below their typical clinical dosing. We see a very, very strong response there. Again, we do see elimination with CAP-B101, but that's kind of as expected. The really interesting data comes in this pre-treated model. So here, this model had undergone 2 prior lines of therapy before extraction. And what you see here is that there is no longer sensitivity to Trodelvy. There is basically no differentiation versus the untreated control. And this model also had been exposed. One of the prior lines of treatment is a Taxol therapy. It's a paclitaxel. This patient had been non-responsive to paclitaxel as well. So we have this patient which was non-responsive clinically to paclitaxel, and where the PDX model is now unresponsive to Trodelvy. And so the fact that, again, this combination approach with this very stable conjugation that we've been able to generate is able to eliminate these tumors is, for us, really exciting. I think really validates this thesis that a stable conjugation of a combination going after different mechanisms is able to overcome some of the resistances that are particularly pernicious in a lot of the patients that we see with these tumors. The next question for us was scalability. Can we scale up the production of these assets? So with a third-party CRO, we were able to scale this process over 1,000-fold. We now have repeatable multi-gram scale production capabilities in industrial stage bioreactors. And we were able to achieve this with absolutely no deviation in overall mAb titers versus the standard wild-type antibodies. And so we see that there's a very, very clear pathway to scalability, particularly given the remarkable purity of the products we're able to achieve. So as I mentioned before, we're doing minor adjustments to the reaction conditions to block access to the different sites of modification. We see virtually no detectable off-target modifications and no increase in DAR of each of our payloads. So the DAR is precise, the sites of modification are precise. We generate a truly homogeneous product in a very scalable manner. And so this for us has been really exciting validation of the approach we've been taking to production here for these ADCs. The next and most important question is, do we see a strong safety profile, and do we see good stability? So our half-life, we do see a very nice improvement in total ADC, almost doubling the established half-life for Dato-DXd. And really exciting for us was that we saw that this completely eliminated the GI tox that is hallmark of, apologies, can I go back? Oh, there we go. We saw almost complete elimination of the GI tox that is a hallmark of both the auristatin and the topo-1 inhibitor class. And we also saw that bone marrow tox was dramatically reduced. So this for us was really exciting. Again, very validating that this approach is generating a very tolerable and very effective molecule. Overall, when we think about the overall therapeutic window of these assets, here we're looking at a comparison of the tolerability versus the ED50 level for CATB101 versus Dato-DXd and SG. And we see a nice improvement in overall therapeutic window, which we think is, again, emblematic of what we'd like to see. A very effective molecule that also has an improved safety profile. Generally speaking, we do think NSCLC is a really exciting target space for this molecule. That's an indication where we've seen time after time failure of topo-1 based ADCs, and there's clearly a sensitivity to tubulin inhibitors based off of the dominance of docetaxel versus some of those topo-1 ADCs. And so that's a space that we think is really exciting for this molecule. There's also some potential in esophageal and gastric cancers, as well as some other tumor types, including TNBC. And that overall, I think, is, again, because of this combination approach, we expect to see greater durability and hopefully greater depth of response in patients. Overall, we've developed a really exciting pipeline. This is just the lead asset that I've been talking about mostly today. We have that HER2 molecule, which I think is a great proof of concept, and we're developing other assets with, against other targets and with other combinations as well. We've also been able to conduct a variety of work with some fantastic third parties. And so with some major pharma groups, we've been able to successfully attach protein cargo to antibodies, which we were able to successfully deliver in vivo. We showed we were able to achieve on-target delivery of those proteins. Another partner we worked with wanted to access unique geometries for single binding domain proteins attached to each other. We were able to achieve that and scale it up quite successfully. And then finally, with our third pharma partner, I think this really exemplifies the flexibility of this conjugation. We were able to take viral capsids and attach single binding domain proteins to various sites on the surface of these capsids. And we found that we were able to titrate this modification to any desired level. Anywhere between 5 to 95% of the capsid was modified with these single binding domains. This is really important because if you have to express that capsid with the binding domain fused, you typically see a fairly significant reduction in overall viral yields. And so this approach, we think, is a really enticing way to add targeting capability to these gene therapies. Overall, I hope I've left you with a very exciting conjugation approach that allows us to attach multiple payloads stably to antibodies with absolute site specificity, a tunable DAR, high manufacturability, and very broad flexibility, not only for payloads, but also modalities in general. I've been very fortunate to work with a phenomenal group of scientific pioneers and very, very seasoned oncology professionals, as well as an amazingly talented group of scientists and fantastic supportive investors. I'd be very happy to answer any questions. Thank you all so much for the time. Microphone?
Harshal Nandarkar from Australia, Melbourne. Great talk. How many molecules of payload can you attach to each antibody? Do you have a perfect control, or is it all dependent upon those residues, individual 2 molecules of residues that you change?
So we have flexibility to attach anywhere from 2+2, 2+4, as I focused on today, all the way up through 6+8. Our thesis here at Catena is we want the minimum viable DAR. So our goal is to attach as few molecules as possible to still show therapeutic improvement. And so we went through a large screening process that I didn't want to include in today's discussion because that would have eaten up a large portion of my time. But we found that 2+4 ratio was quite optimal to deliver therapeutic doses of both the tubulin and topo-1 inhibitors. But we do have greater flexibility beyond that ratio. We can accommodate a variety of different combinations of payload ratios.
Thanks.
Are you thinking about any other dual payload combinations beyond the two that you just described?
Yeah, absolutely.
I mean, I know you talked about the protein antibody, but I guess I'm thinking more on the cytotoxics.
Yeah, absolutely. We've looked at a variety of some of the cytotoxics here. I think the question for us is this improvement in off-target tox enough to enable some of the historically more challenging payload classes? I think the jury's still out on whether more stable conjugation is enough to overcome some of those barriers there. We are certainly actively evaluating. We have some that we're very excited about internally, but we haven't talked about those publicly yet.
Yeah, so it's really a question of whether the benefits will outweigh the risk.
That's exactly it. And I think importantly, are you able to match potencies, right? If you have something that's 1,000-fold more potent, you're not going to be able to deliver enough of any of the partner molecule to actually generate an active dose for that. And so I think that's one of the biggest limitations when we think about the dual payload space is, do you have two molecules that are close enough in potency that when they're delivered on the same molecule, they are actually both going to be active? That I think is one of the biggest limitations beyond even, I mean, there's certainly a quest in ADC land for more varieties of payloads, but I think if you're going to combine them, you have to ensure that they're near match in potency. Otherwise, you're going to be generating a molecule that only really shows activity of your most potent payload.